Hello dear friends, welcome back. In this video, we'll be discussing GATE MCQs and MSQs of Biochemistry paper 2024. We'll be solving question 28 to question 32, each carrying 1 mark. Let's discuss. Question number 28. Which one of the following pairs of amino acids is not incorporated in a polypeptide chain? Options are 4-hydroxyproline, gamma-carboxyglutamate, gamma-carboxyglutamate and desmosine, ornithine and citrulline, 4-hydroxyproline and 5-hydroxylysine. The answer is ornithine and citrulline. Let's see the explanation. Here is the explanation. Role of ornithine and citrulline are involved in metabolic processes like urea cycle, vasodilation, and nitric oxide synthesis. Ornithine and citrulline are not found in protein structure of living organisms. On the other hand, 4-hydroxyproline and 5-hydroxylysine are involved in collagen synthesis and structure. Let's go to the next question. Question number 29. Mammalian cells cultured at low temperatures, that is 25 to 30 degrees Celsius, leads to an increased sterol content in the membrane. Elevated sterols in the membrane results in options are an enhancement in membrane fluidity, stabilization of membrane proteins, an increase in membrane permeability to water, a decrease in membrane fluidity. The answer is an enhancement in membrane fluidity. Let's see the explanation. Here is an explanation. At low temperatures, fatty acid chains become more packed and rigid. Due to an increased rigidity of the membrane, there will be a decrease in membrane fluidity. Once the rigidity in the membrane is increased, the membrane fluidity decreases, due to which various cellular processes like signal transduction and protein translocation would not occur easily. So to balance for an increased rigidity, cells will either increase uptake of sterols from the surrounding medium, or they will increase synthesis of sterols within the cell to maintain membrane fluidity. Sterols like cholesterol prevent cell membrane from freezing by interfering with fatty acid chains. Let's move to the next question. Question number 30. Which one of the following metabolic intermediates is common to glycolysis, nucleotide synthesis and glycogen synthesis? Options are citrate, oxaloacetate, glucose 6-phosphate, glycerol 3-phosphate. Let's see the explanation. In glycolysis, there is an isomerization of glucose 6-phosphate to form fructose 6-phosphate, which is catalyzed by an enzyme phosphoglucoisomerase. In nucleotide synthesis, glucose 6-phosphate acts as an intermediate in pentose phosphate pathway, in which ribose 5-phosphate and NADPH are generated, which are further utilized in nucleotide synthesis. In glycogen synthesis, isomerization of glucose 6-phosphate to glucose 1-phosphate catalyzed by an enzyme phosphoglucomutase. So in this case, all three pathways using glucose 6-phosphate as an intermediate. Let's move to the next question. Question number 31. In mammals, hematopoietic stem cells that give rise to different types of blood cells are known as. Options are totipotent stem cells, pluripotent stem cells, myeloid progenitor cells, and lymphoid progenitor cells. Let's see the explanation. Totipotent cells. These cells have ability to produce all the differentiated cells in an organism, including embryonic and extra-embryonic tissues. Pluripotent cells. These cells have ability to form all the derivatives of the three germ layers, that is ectoderm, endoderm and mesoderm, but these cells exclude, that is extra-embryonic tissues, the placenta and the umbilical cord. Myeloid progenitor cells give rise to polymorphonuclear cells or the granulocytes, that is neutrophils, basophils and eosinophils. Monocytes, macrophages, dendritic cells, osteoclasts, mast cells, platelets, and RBCs. Lymphoid progenitor cells. These cells give rise to T cells, B cells, natural killer cells. So the answer is all types of blood cells are formed from pluripotent stem cells from the mesoderm germ layer. Let's move to the next question. Question 32. Which one or more of the following statements correctly describes the addition of N nucleotides during the rearrangement of the immunoglobulin heavy chain encoding gene? Options are: addition of N nucleotides is template encoded. N nucleotides are added by terminal deoxynucleotide transferase. The added N nucleotides are common in VD and DJ junction. N nucleotides are added by DNA polymerase II. To, to this question, the answers are, this question is MSQ, that is multiple selection question. So there are two options. The correct options are N nucleotides are added by terminal deoxynucleotide transferase, and the option is second option is C. The added N nucleotides are common in VD-DJ junction. Okay. Option A is eliminated because addition of N nucleotides is template independent manner, and option D is also excluded, that is N nucleotides are added by terminal deoxynucleotide transferase, not by the DNA polymerase II. Here is an explanation. In a developing B cell, DNA rearrangement occurs through a random combination of VDJ gene segments by the process called VDJ recombination. During joining of the gene segments, variable number of nucleotides are often lost from the ends of recombining gene segments, and one or more nucleotides are randomly inserted. This random lose or gain of nucleotides at joining sites called junctional diversification, due to which diversity of V region coding sequences increases by recombination. N nucleotides added at the DNA end are random and template independent manner, and the enzyme, that is terminal deoxynucleotide transferase, adds 15 N nucleotides at DNA ends. Here is the end of the question. See you in the next video. Thank you.